Hello everyone and welcome to our video on anaerobic respiration in eukaryotes. This is part of our Module 5 respiration series of videos, and in this particular one we'll be having a look at specification reference 5.2.2i part 1, the process of anaerobic respiration in eukaryotes. So what we've been looking at over the past few videos is that aerobic respiration process in eukaryotes. Now, obviously aerobic respiration is a process that can take place when we've got a plentiful supply of oxygen, because that means we've got the oxygen available to be that final electron acceptor in the process of oxidative phosphorylation. A problem arises when the organism has a lack of oxygen, because in that scenario what it means is that we don't have oxygen as the final electron acceptor in oxidative phosphorylation. We won't be able to form water from the protons that diffuse through the ATP synthase channels. The concentration of protons will therefore increase in the matrix. That's going to reduce the proton gradient across the inner membrane of the mitochondria, and therefore oxidative phosphorylation stops. If we're not able to maintain that concentration gradient of those protons, then they won't diffuse through the channel, and therefore the whole thing just grinds to a halt. So this lack of oxygen is going to bring that process to an end. The key thing to remember though is that even when we have a lack of oxygen, we can still carry out glycolysis. So that very first part we looked at in our aerobic respiration pathway, the process of glycolysis can still take place because there was no oxygen requirement in that part. When we think about what happens in glycolysis, hopefully we remember that we're producing reduced NAD. Now, in order for glycolysis to continue, we need, obviously, oxidized NAD. We need the NAD that can become reduced. So we've got two options available as to how to achieve this, and the option depends on what organism we're talking about. If we're talking about fungi and plants, they will use a process called the ethanol fermentation pathway to reoxidize that reduced NAD. If we're thinking mammals, it would be the lactate fermentation pathway. So two different pathways to reoxidize that reduced NAD, and it's determined by what organism we're actually talking about as to which one would be utilized. Let's start off with the fungi and plants then. So we're talking ethanol fermentation first of all, and this is going to take place in the cytoplasm of the cells. Now what we see happening here, remember, is we've had glycolysis take place, and therefore we do have our pyruvate. Now pyruvate is then going to be decarboxylated. So remember, decarboxylation is where we're going to end up removing the carbon dioxide here. So it gets decarboxylated to form ethanal. Go careful with the ending. Now in order to carry out that process we need an enzyme, and the enzyme is pyruvate decarboxylase. So obviously we're decarboxylating, so it's a decarboxylase, and the thing we're decarboxylating is pyruvate. The ethanal is then going to accept hydrogen atoms from our reduced NAD, therefore reoxidizing it into our NAD that we needed to reform, and those hydrogen atoms form with the ethanal to make ethanol. And this again has its own enzyme, this time ethanol dehydrogenase, because we are carrying out a dehydrogenation, because what we're doing here is obviously changing our reduced NAD into NAD, so we're oxidizing, and that means we are removing hydrogens from it. So two different enzymes involved. Go careful with the enzymes that we're talking about, but the end product here is obviously ethanol, hence it's called the ethanol fermentation pathway, and we've reoxidized to our NAD, which can then go back to be used in glycolysis once more. So this one here just gives you a little overview of the process we've gone through then. So we start with our pyruvate, which is the end product of glycolysis. That is going to undergo the decarboxylation. So we're going to form carbon dioxide, and that's in the action of pyruvate decarboxylase, and that means our pyruvate becomes ethanal. What we then find the second stage to the process is our reduced NAD is reoxidized into NAD. The two hydrogen atoms that are removed will then be attached to our ethanol to make ethanol. And that's through the action of our ethanol dehydrogenase. So make sure you go careful in terms of which enzymes occur at which point of the process, and don't mix up your ethanols and your ethanols. If we now consider the process in animals, we're talking lactate fermentation pathway, still occurring within the cytoplasm of our cells, and we typically find this occurring within the muscle tissue during vigorous activity. So once more, we're starting with pyruvate, because remember, glycolysis has taken place in all of these cells, therefore the end product of glycolysis is pyruvate. Now that pyruvate is going to be reduced to lactate, which is our end product here, and it's reduced by accepting hydrogens, which are obviously going to be removed from the reduced NAD, which is reoxidized to form NAD. So what we've got here, reduction of the pyruvate, oxidation of the reduced NAD, and it all takes place in the presence of lactate dehydrogenase, because obviously we're removing hydrogens there, therefore we're going to take the hydrogens from the reduced NAD to reoxidize it. Those hydrogen atoms then combine with the pyruvate to make our lactate. When we think about lactate, then we have a bit of a problem, because when that lactate accumulates within the muscle tissue in our mammals, for example, then it's going to actually lead to a decreased pH. So the pH is going to get lower. And the problem with lowering pHs is that that has an impact on enzyme activity. So what we'd actually find is that this accumulation of lactate, lowering the pH, would actually end up inhibiting the enzymes that are involved in any of those other processes happening in the area, like glycolysis or muscle contraction. So to try to minimize these effects, what we find is that the lactate is transported in the blood to the liver. So we're going to basically move it away from the area to avoid that pH dropping to those problematic levels. Then, when we get more oxygen available, there's two options for what's going to happen to the lactate. Option one, it gets converted back into pyruvate, and at that point enters the link reaction and off we go. Or it could be recycled to glucose and into glycogen. So we do have those two options available to us as to what we do with that lactate at the point that we have more oxygen available. So remember, we sort of said this tends to happen during strenuous exercise, and therefore if exercise is ended, what we're going to see is one of these two things will take place. One thing that we've been focused on over these past few videos is the production of ATP. And it's really important that you remember that neither the ethanol fermentation nor lactate fermentation pathways actually produce any ATP. So the reason that organisms continue to get some ATP is because of glycolysis. It's not due to either of the fermentation pathways, because they produce no ATP themselves. It's that they enable glycolysis to continue, because what they're doing is reoxidizing that reduced NAD, which can then obviously allow the glycolysis process to carry on. So what that means is, if we're thinking about our net yield of ATP in anaerobic respiration, it's a grand total of 2 ATP molecules per glucose. A little bit different to the 32 we looked at with the aerobic. So, as always, I do suggest you come up with these simple tables where we get these two similar processes where they do have certain aspects that are in common, but there's also the differences. They lend themselves very well to comparison questions on an A-level paper. So creating yourself a little table like this that's going to help you to obviously recall those key differences between them means should that style of question come up on your exam paper, you've got that quick recall that you can use and either fill in a table they give you or give yourself a nice written answer. Couple of points about those kinds of questions though before we move on to look at the table itself. If they give you the outline of a table and ask you to do a comparison between the two fermentation pathways, you must make sure that on any given row that you are making the direct comparison. So on the top row there, we'd be putting down about the hydrogen acceptors. So we would say ethanol as a hydrogen acceptor in our ethanol pathway, and then next to it, we would have pyruvate as the hydrogen acceptor for the lactate fermentation. If you're not putting them as a direct comparison on a row, you're not getting the marks. We're not going hunting for where in the table they might correlate. It's got to be in that same row. So go careful with the tables. If it's a written answer that we're doing, then you've got to make that clear comparative statement. You couldn't just say carbon dioxide is produced in ethanol fermentation. That's not a comparison, that's just a random statement, folks. You'd have to say that carbon dioxide is produced in ethanol fermentation, but not in lactate fermentation. So it might sound really quite pedantic at times, but we are asking you to compare two processes. If all you're doing is making a point about one of them, that's not comparing. So make sure that on any of these comparison questions, you are adhering to those guidelines. Otherwise, you're throwing away really straightforward marks just because of how you're setting your answer out. Okay, quick recap of our little table here then. Ethanol fermentation on the left, lactate fermentation on the right. So we do have that different hydrogen acceptor. We do have a difference in whether carbon dioxide is produced or not. Similarity that neither produces ATP there. Obviously, different end products, as we can see in the table, and different enzymes will be involved in that. Similarity in terms of the fact the NAD will be reoxidized in both cases, and the similarity that they're both taking place in the cytoplasm. So we've got similarities, we've got differences. As I say, really nice comparison question that could come up on papers there. As always, I hope you found this video useful, and of course don't forget to subscribe to the channel so you can see when further videos are uploaded, and head on over to the A-Level Biology website where you can find a range of other resources to assist you in your A-Level biology studies.